Good morning, everyone, or evening. Hope y'all are having a good day. So today we're going to learn about the bilipid membranes, coronavirus, and hand washing, and kind of figure out how they all even go together. So before we begin, please pause this video while you go and grab your pen and your science notebook. All right, so now that you have your science notebook and you're ready to go, we're going to really quick go over a couple things that we've been talking about for the last couple weeks. So we have discussed the cell structure and how all cells have a cell membrane, which is shown in orange on this diagram. They also have cytoplasm, the grayish blue fluid that fills up the cell. And it has genetic material in the nucleus. If it's a prokaryote, it would be in the nucleoid. And they all have membrane-bound organelles if they're a eukaryotic cell. We are going to mainly focus on the cell membrane today and talk about some of the chemical components of the cell membrane. And they include the ideas of polar versus nonpolar. Now remember that nonpolar substances are like lipids, so they're oils and fats. And we have polar substances, which is like water, carbohydrates, and proteins. And we've heard a bunch of times that like dissolves like. So nonpolar things will dissolve in nonpolar other substances, and then the same thing for polar materials and substances. Also, we can think about it as polar molecules are super clicky and like to be only with polar molecules, and that nonpolar molecules are super clicky and only want to be with other nonpolar molecules. So when we go and we look at the cell membrane, we are looking at a bilipid membrane structure. So that bilipid membrane is going to be made up of phospholipids. A phospholipid has a hydrophilic or water-loving head, which is this kind of reddish circle. And then it has hydrophobic or water-fearing tails, and those are those fatty acids that are coming off of that circle. Due to the phospholipid structure and the clinginess of polar and nonpolar molecules, they arrange themselves to form a bilayer. In this bilayer, we have that the lipids, the fatty tails, are going to arrange themselves to where they're all going to only be near other fatty tails. And we're going to have the hydrophilic heads with the phosphate groups on either side of that membrane. So this structure ends up being very, very strong protective barrier for cells and other things with membranes to protect it from hydrophobic and hydrophilic substances from going in and out of the cell unless they have a special transport method. So if they're so important, where are they found? We know that all living things are made of cells. And all cells have a bilipid cell membrane, as you can see in this diagram. It shows it going all the way around in that yellow and orange. We also know that eukaryotic cells have membrane-bound organelles such as mitochondria, chloroplast for plants, and a nucleus that holds all the important genetic material. We also know that substances and particles do have to enter and exit a cell. We need to get nutrients into the cell, and we got to get waste out of the cell to keep it healthy. So they also have vesicles that are little spheres of just bilipid membranes that can help transport materials in and out of cells. So what's this got to do with COVID-19? Well, let's look at the structure of a virus, because a virus isn't really a living thing, but I think we'll notice there's a lot of similarities of the virus structure to a living cell structure. So viruses have a protein capsid. So proteins are polar, and it's this little diamond geometric sort of shape. And then within that capsid is where we have the viral genome. So all of the virus's genetic material. And then some viruses, like COVID-19, have something called an envelope. The envelope is the virus's own sort of cell membrane. So the virus, when it's leaving its last host cell, will borrow or take some of the bilipid membrane and use that for themselves to make a protective barrier called the envelope. You can see in step 4 of this diagram at the bottom, when viruses are leaving that host cell, you can notice that they do have that envelope as they leave, but they don't have it when they're in the cell. And that's how they get out. So now for hand washing. But first, let's do an experiment. So be ready to jot down some hypotheses and observations. So I have with me just 3 simple things that you can have in your own house or apartment, wherever you are. So I have extra virgin olive oil, any oil or lipid will do, a mason jar full of water I just filled up from the tap, and then I have some zesty lemon hand soap. Again, any soap will do. And I want all of you to take a moment, pause the video, and write down what you think will happen when I add the olive oil into the mason jar with the water. Okay, so I think you can all notice that this mason jar now has a layer of oil on top and the water on bottom. But I poured it really carefully, so maybe if I mix it or something, it'll do something different. So while I'm mixing this, write down what you think will happen after I've given this a good shake. Right. Let this settle. Well, I'm already noticing that the oil is immediately separating again and I have the water on the bottom. So we can think of this space right where the oil meets the water as a cell membrane or an envelope for the virus. So now we're going to add some soap and give it a good shake and see what happens to this envelope, viral envelope. Here we get a nice, good amount of soap. Right. Shake this up again. Okay. We notice there's no separating happening anymore. That viral envelope was completely taken out just by a little bit of hand soap. Yeah, there's no separating at all. All right, so what just happened? Let's take a look. So we added soap and it's an amphipathic molecule. So that means that this molecule has both a hydrophobic and hydrophilic region. So if we see, this is a typical structure of soap, and we see that it has this hydrophilic head, and then it has a long tail of a bunch of things that are hydrophobic or nonpolar. Kind of looks like a phospholipid if you think about it. And they're going to interact with bilipid membranes like viral envelopes differently than hydrophobic and hydrophilic molecules. they are going to almost pull apart that envelope because it has some part of the molecules that are going to be attracted and be able to click with the polar part of the membrane, and it's going to have other parts of the molecule it's going to be able to click and be attracted to the nonpolar parts of the membrane. So that's why we still look at this. It still hasn't separated at all. And that's because the envelope was destroyed by that amphipathic molecule. And now it takes about 20 seconds for soapy water to destroy a viral envelope, which is why they always say you should wash your hands with soap and water for 20 seconds. So let's do a quick recap. What we learned today. So we know that the bilipid membranes, often a phospholipid bilayer, are made of phospholipids that orientate themselves into a strong protective barrier. We've also learned that some viruses, like COVID-19, steal some of the phospholipid bilayer for themselves to make their own viral envelope. We know that soap is an amphipathic molecule, so it has hydrophobic and hydrophilic regions. And an amphipathic molecule, like soap, can destroy a viral envelope in about 20 seconds. So the moral of the story is wash your hands, destroy some phospholipid bilayer, and stop the spread of COVID-19. And some reminders, before next class, please make sure you complete the Google form that covers the material we covered today in this lesson. And we also have questions written down in your lab notebook that we can discuss next time we see each other. All right, hope you have a great rest of your day. Stay safe.